All right, better get going. Hi, everybody. I'm Soleille Miller. I'm a PhD student at UNSW in Sydney, Australia. And today I'm really excited to be able to share with you guys a little bit about the work I've been doing throughout my PhD. So this will be a bit of a synopsis of pretty much my entire thesis. So, why do most animals reproduce sexually? Theoretically, asexual reproduction is just more efficient. As an asexual, you don't have to put any resources creating males that can't then go on to reproduce themselves. All else being equal, this creates a twofold cost of sexual reproduction. In addition to this, as a sexual organism, you need to spend valuable time and energy seeking a mate, at which point you are exposing yourself to a greater risk of predation. Not to mention, once you do secure a mate, you have to deal with the many physical costs that are often associated with mating. So then, if sex is so costly, why is it so common among the animal kingdom? So as it turns out, asexual reproduction comes with its own set of costs, most notably the loss of genetic diversity. But also, a lack of meiotic recombination results in extensive linkage across asexual genomes. And this means that the deleterious and the beneficial alleles cannot be acted upon independently by selection, resulting in less efficient selection, as well as an accumulation of deleterious alleles, ultimately reducing the fitness of asexual organisms over time. These long-term costs often mean that asexual lineages face an early extinction, ending up at the short end of the phylogenetic stick. But not always. Some animals are really, really good at asexual reproduction and have been reproducing asexually for millions of years. So theoretically, this means that asexual reproduction can be a viable long-term strategy. But what separates the rotifers and the ostracods from 99% of all other animals on Earth? So I tried to investigate some of these broad-scale evolutionary questions by looking at the initial stages of transitions from sexual to asexual reproduction, to try to identify some factors that could play a role in the success or failure of asexual lineages in the wild. And I do this by studying an animal that is able to reproduce both sexually and asexually. Animals with this ability are known as facultative parthenogens. And when they do not mate, they will reproduce asexually, not fertilize their eggs, and have only daughters. But when they do mate, they will reproduce sexually, fertilize their eggs, and have an equal sex ratio of males to females. Now, recent work suggests that this strategy is not evolutionarily stable. Instead, it's a transient state in the evolution of complete asexuality or complete sexuality, and the scales are always going to tip one way or the other. This makes facultative parthenogens a really great system to look at those initial consequences of transitions to asexuality as they unfold in real time. So here's where I introduce the star of the show, the focal species of my PhD, Megacrinium batesii, otherwise known as the peppermint stick insect. So these are facultative parthenogenic insects, very charismatic critters from the wet tropics of northeast Queensland in Australia. They're pretty big. They're about this big, pretty large for insects. Makes them very easy to, very accessible in the wild, easy to find, makes them great for fieldwork studies. They're also pretty easy to rear in the lab, making them really good for lab work studies. And in my PhD and in my lab, we do a bit of both. So today I'm going to be talking mostly about the consequences in the wild. So in their natural range, this reproductive flexibility creates a mosaic of distinct sexual and asexual populations that are sometimes in really close proximity to one another, yet they are distinct populations. And within this range, they can be found in two main contrasting environments. Either lounging beachside on the pandanus houseplants, where they're more exposed to the sun and wind, or in the closed canopy rainforest swamp habitats, where they're on Bensonia houseplants and are less exposed to the sun and wind, but perhaps have more access to fresh water. So, both sexual and asexual populations across the range can be found in relatively equal frequencies across both habitat types. This is going to come into play a little bit later, so keep that in mind. So in my first chapter, I discovered at least four independent transitions from sexual to asexual reproduction within the natural populations. And throughout the rest of my PhD, I really dive into these transitions to try to understand a little bit more about the early consequences of transitioning from sexual to asexual reproduction. And I do this by looking at 3 different perspectives. I look at the environmental differences, the genetic differences, and the phenotypic differences between the reproductive modes. Very quickly, I'm not going to spend a whole lot of time on the environmental differences because, in short, I didn't really find any. I'm actually going to spend more time talking about the genetic and phenotypic consequences. So, in terms of their genetics, we can see, just like I said earlier, the asexuals do seem to have really low heterozygosity values compared to their sexuals here in purple. However, there's a bit of variability in this. And I found a few individuals with heterozygosity values more consistent with sexual reproduction, despite no males being found in these populations over 5 years of surveys. But again, there is a pretty drastic difference between the 2 reproductive modes in general. We don't know how long these individuals have been asexual in the wild, definitely more than 5 years, but past that, we're not entirely sure. So we don't know how quickly this heterozygosity erodes following a transition. So that is exactly what I investigated next. And to try to understand how quickly heterozygosity erodes over time, we took sexual individuals from the wild and ran them through 2 generations of asexuality in the lab. This was work done by my lab mate Daniela. This dataset was generated originally to investigate sexual conflict, female resistance, and the decay of sexual traits. So please, if you get a chance, please go see her talk. It's in the next session. She put a lot of work into it, and she's got some really cool results. So please go see her talk. But for my part of the project, I took the insects from a project and sequenced them and followed them through these generations to see just how quickly that heterozygosity is lost. And I found that it typically takes only 1 generation after transition to asexuality to lose most of their heterozygosity, down to that baseline level that we see in the wild. And then they stay at that baseline for the 2nd generation. This is most consistent with a specific mechanism of parthenogenesis, known as automixes with gamete duplication, which results in really low to 0 heterozygosity in offspring. However, just like in the wild, I did find a bit of variability in this. And I had a few individuals that had heterozygosity losses more consistent with 2 other mechanisms of parthenogenesis. So this suggests that there is some variability in the wild populations that can manifest itself as diverse mechanisms of parthenogenesis when they transition to asexual reproduction. Now, despite the drastic genetic differences I saw between the 2 reproductive modes, this didn't seem to translate into any clear phenotypic or fitness consequences of transitions to asexuality, at least not in most of my traits. I did find that there was a difference in terms of wing deformities, where asexuals did have higher numbers of wing deformities, but wings are non-functional in microcrania. They're a little bit too chunky for the wings to actually help them. So it's unlikely that these wing deformities actually have any sort of effect on their performance or fitness in the wild. However, ectoparasites tells a slightly different story. So here, by ectoparasites, all I meant was the visibility of fungal infections or visible invertebrate ectoparasites found on the insects. And as you can see, across the different populations, there's a bit of variability, but there was no general difference between sexuals and asexuals. That is until we separate them based on habitat type. In the beachside pandanus habitat type, we can see that there isn't a difference in terms of ectoparasites between the sexuals and asexuals. However, when we look at the swamp habitat, we can see that the asexuals are more likely to have ectoparasites found on them than the sexuals, but again, only in that habitat type. So this suggests that there may be differences in selective regimes between the 2 habitat types that's affecting these insects. So this is the next thing I investigated a little bit further by taking my sexual individuals and doing the genetic data for my sexual individuals and doing a selection analysis using BayPass. And using VIII, I identified 11 putatively selected alleles that are significantly associated with habitat type in these insects. And I found that individuals from the swamp habitat were more homozygous at these outlier loci. So that's the top left figure up here. They're more homozygous at these loci compared to their sexual conspecifics. And this pattern was not evident when looking at the neutral loci on the top right for you. In addition to this, I did a PCA using these 11 loci and found that the individuals from the swamp habitats were also more genetically similar to each other compared to the sexual individuals, the beachside individuals. And again, this pattern was not evident when looking at the neutral loci dataset. Instead, they just clustered based on geographic location. So taken together, this suggests that there's stronger selection on these insects in the swamp habitat, and this could explain why why we're more likely to see ectoparasites on the asexual individuals in the swamp habitats, where the reduced efficacy of selection in the asexuals may be a disadvantage for them, and they might not be able to keep up with the challenges that this specific environment brings. So overall, I found no general environmental differences between reproductive modes, but I did find a drastic and immediate loss of genetic diversity following a transition to asexual reproduction. However, this varied based on the cytological mechanism of parthenogenesis, of which there are at least 3 within my populations, which provided a little bit more genetic variation. Now, despite this dramatic genetic difference, I don't see this translated into any clear, overarching phenotypic or fitness consequences of these transitions. However, this again varied based on the environment that they were found in. So ultimately, I've determined that the success or failure of asexual lineages is likely to be highly context dependent, which I'm aware isn't the most satisfying conclusion to a presentation, but when you're trying to answer big, broad, evolutionary questions like why is sex so common in animals, the answer is often elusive because, like most things in nature, it's complicated. Thanks, everybody, for listening to my talk. Special thank you to the Rolling Stones and Bond Lab, some of which who are here today. And yeah, a special shout out to Kat Stewart, who has helped me a lot with this. And yeah, happy to chat about this or anything else. Please come up, talk to me, ask questions. Happy to chat all the time. Thanks for listening. Yes, so they can. We've actually, funny enough, we've got one population. Oh, it's really far back. We'll just stay here. We've got one population that seems to be in the transitionary period back to sexual reproduction. So they've actually been excluded from all of these analyses, unfortunately. But yeah, we do see that in the wild that can happen, but we're not entirely sure what the consequences of that are going to be. Because it just happened like 2 years ago, and so each year we follow up on it, and it seems like the males are starting to invade, and right now we're almost at a 50-50 sex ratio in that population. So yeah, one instance. Yes?
I might have missed something, but it seemed like you discounted environment at the start, the general environment, but then you show at the end that there's actually like a really big impact of environment.
Yeah, so I didn't talk specifically about what I meant by the environmental differences. So generally, they're at a very small range. It's like about 35 kilometers total. So in terms of general broad scale temperature differences, it's unlikely to affect it too much. And for my second chapter, I actually went into the wild and tried to look at some more smaller scale differences, specifically in terms of host plant niche differentiation. And I didn't find any differences in terms of host plant niche differentiation. Also, in terms of the beach and swamp, I don't know which one that is, but I'm going to talk for maybe a tiny bit more. In terms of beach and swamp, they are like relatively evenly spread across it. It was just that one phenotypic trait, the presence of ectoparasites, that we did actually find an interaction. So it's not to be completely thrown out, and there's definitely more to search for there, especially more micro habitat differences. But in the thesis, I haven't been able to do like the smaller scale things. I've done mostly niche differentiation. Yeah. All right, I'm gonna get out of here. Thanks, guys.
Okay, hi everyone. I am Swank. I'm a PhD student in Dr. Sarah Davies' lab at Boston University. Today I'm going to be talking about some of the research I'm doing for my dissertation about depth specialization driving coral cryptic lineage distributions. So corals, as most of us in this room probably know, are very complex organisms. So you have this coral colony that can be made up of hundreds to thousands of individual polyps. And these coral colonies all have unique genotypes. So in a population, we know that having a diversity of genotypes enables a population to be more resilient to changes in environmental conditions. But corals are really complex because they also have these symbiotic algae and bacteria that also have their own genomes. So these algae also have unique genotypes that all interact with one another, and then these unique genotypes also interact with the host genome, and that makes predicting these responses to environmental conditions very challenging. And one particular environmental stressor that we know is affecting corals greatly is thermal stress. So increases in temperature threaten this relationship of the corals and their symbiotic algae. But what is interesting is that recent increases in genomic techniques have uncovered a lot of this cryptic diversity that we didn't really understand to begin with. So a lot of times you see corals that look very morphologically similar, but they're actually in different taxa, and sometimes they look very different and they're in the same taxa. So we see these patterns across genera in corals where we have these emerging cryptic diversity situations happening. So all of this poses a really great question that we all wish we knew the answer to, which is, what is a species? Everybody can have their own opinion. I'm just in the first year of my PhD, so I'm not here to tell you what that is. But what I can tell you for this talk is that we have this situation somewhere between a population and a species where we have this cryptic lineage divergence. So cryptic lineages are morphologically similar but genetically distinct groups. And so we found in this recent publication by a collaborator in our lab, this is a perspective piece where he talked about cryptic lineages and things that separate them. And typically when you find a cryptic complex, you see that it's segregated by some sort of environmental variable. So if we use depth as an example, you might find a cryptic lineage like this blue one that lives in shallower depths, and then you have on the other end this yellow lineage that lives in deep depths, and then somewhere in between you might have generalists or other cryptic lineages in there. And so in the Davies lab, we're really interested in this species complex Siderastria siderea in Bocas del Toro, Panama. Siderastria are a bouldering coral, so they're broadcast spawners. And in a previous study done by Hanno Eichelman in our lab, they collected Siderastria from offshore sites and inshore sites and found that there were 3 distinct genetic clusters where we had one lineage. Lineage 1 is our offshore lineage. Lineage 2 we found mostly inshore. And then there was a 3rd lineage that we rarely collected but was also inshore. And so when we look at the phenotypic differences in these lineages, we see that lineage 1 appears to be more resilient to heat stress. So in this figure, on the x-axis, you have days of an experiment, and the y-axis is FV over FM, which is photosynthetic efficiency. It's a common variable collected to understand the health of coral throughout a bleaching event. So when we look at this figure, we can see that lineage 1 was largely resilient to this heat challenge, whereas lineage 2 did not do well. But what we didn't collect when we collected these samples was depth data. So it's possible that we put these corals in this experiment and lineage 1 came from environments where they had higher light availability. And so that could have been an impact in this experiment where we exposed them both to the same light conditions, which might have been more ideal for lineage 1, but not for lineage 2. And this is further supported by data on a few samples where we collected information on the symbiont composition. So here we show that lineage 1 hosts more of the genera Durastinium, and Durastinium has been shown to be more resilient to these thermal stress conditions compared to Cladocopium, which is less resilient. And so for my dissertation, I'm interested in looking at how host and symbiont genetic diversity in this cryptic species complex can be shaped across depth gradients. And so we went back to Bocas del Toro reef complex, and we collected 167 Siderastria across the full depth gradient and collected depth information with each sample. And we used 4 inshore sites because we knew that we could find all 3 cryptic lineages at the inshore sites. And then we also tagged all of these colonies so that we were able to return to them in later years and understand how they've changed across time. So this was done in 2022, and we initially sent them out for 2B-RAD sequencing, and we found that there are actually 3 distinct clusters. And when we put these data into a PCA, we see that these 3 clusters are not associated with site. So the different colors of the points here are representative of the different sites that we collected them at. So when we look more closely, we see that these 3 clusters are actually associated with these 3 genetic clusters that we're seeing over here. So we're calling them lineages 1, 2, and 3. And then when we redo this PCA just by colors of the lineage, we see that these groupings are just associated with the lineage and not any sort of mixture between them. And this is supported by the FSTs as well, where we see the FST between lineage 1 and 3 is 0.13, and then between 1 and 2 is 0.15. And then our least diverged group is lineage 2 and 3, which is still a pretty high FST of 0.08. And we also see evidence of very limited to no gene flow in our admixture plot here. So you can see we have all 3 samples, or all 3 lineages were found at 2 of our sites, and at the other 2 we found 2 and 1. So when we look at these data compared to the previous study, we find that these are the same genetic lineages that we detected before. But before, we thought that lineage 1 was mostly offshore and lineage 2 was mostly inshore. But as I said, these were all collected at inshore sites, so now we're like, okay, there's like something else definitely separating these lineages. And so we look at where we collected them along the depth gradient. And so on the x-axis here, you have depth, and these top 2 density plots are showing where we collected all 3 lineages at these sites. And so you can see lineage 1 is almost always in very shallow waters. Lineage 2 is more of our depth generalist, so it's kind of throughout the whole depth gradient of the species. And then you have lineage 3 that is almost always in deep waters. And you can see the morphological variation between lineage 1 and lineage 3 here, so maybe they're not as cryptic as we thought. But what is interesting is when we look at lineage 2. So lineage 2 looks more like lineage 1 when you collect it in shallow water, and looks more like lineage 3 when you collect it in deeper water. But then when we go to our other 2 sites, they were sites that we perceived as more turbid waters, so maybe having less light availability. And we see disruptions in these patterns. So we see lineage 2, which was originally our generalist, is mostly in shallow waters. And then at the other site, we see lineage 2 in shallow waters and lineage 3 in deeper, but less deep than it was before. And so this morphological variation is really interesting when we look back to the previous study where we looked at how there were differences in these corallite morphologies and their ability to scatter light. So you can see when you shine a light on lineage 1, it scatters much less because they have very tightly packed corallites. And then in lineage 2, the corallites are more spread out and deeper, which allows them to reflect the light more. So these were really interesting results that paired well across these 2 studies. And so some future directions that we have with this work is to look at ITS2 and 16S metabarcoding data to understand if there's any sort of relationships between the symbionts that each lineage is hosting. And then we also want to repeat these ITS and 16S measurements after subsequent bleaching events in 2023 and 2024. 2024 hasn't happened yet, I'm just assuming. And hopefully we might find some shifts in these microbial communities after these stressful events. And then we also want to relate these data with bleaching and mortality observations that we've recorded from 2022 to present to see if any lineage is more susceptible to bleaching or mortality than others. And so if you're interested in that first study, we have just released a preprint, so you can find that at the QR code here. And I want to thank everybody that has put work into this project. It's been a really big, long-term project that I now get to work on, and super exciting. And if you also find it exciting, the Davies Lab is recruiting a PhD student to start in the fall of 2025. So if you're interested in working on this cool system, or you find me really cool, that's cool too. You can apply at the QR code here. And with that, I can take any questions.